I moved across the country to San Diego as a freshly minted PhD in molecular biology. I was excited, ambitious, and viewed the move as the start of a long research career. I had been accepted to work in a well-known lab at the Scripps Research Institute and saw this as my opportunity to prove myself. I was certain that I would become famous, at least within the world of molecular pathogenesis. The lab I was to work in was developing a vaccine for Lassa fever virus, a hemorrhagic fever that causes a horrible death due to internal hemorrhaging. My enthusiasm for the project was so great, I may actually have been willing to work with active Lassa virus. Fortunately, that wasn't required. I was to work with a related virus that causes no harm to humans, but does make mice sick. The mouse immune system is very similar to the human immune system. So to test vaccines, you first inject them into mice and see if it generates an immune response. If it protects the mouse against infection, there's a good chance it will also protect humans. All researchers who are going to work with animals must take a class called The Humane Care and Treatment of Animals. I sat in the back of that class, rolling my eyes, thinking to myself, if I saw a mouse in the corner right now, I'd throw a shoe at it. So why the hell am I wasting an hour of my time on this stupid training? The training did not actually feature live animals, just a lecture and a video. So it was easy to maintain that jaded demeanor. The following week, I was finally ready to meet the mice. Farzana, the lab tech who was helping me, took me to the animal facility, a warren of windowless rooms, their walls lined with mouse cages. Before we could enter any of the rooms, we had to first put on protective gear. This meant clean lab coats, latex gloves, paper mask over mouth and nose, paper booties over our shoes. In my mind, that first time in the animal facility, these preparative steps added to the excitement. I was a real scientist, about to go do important work. We entered the room where our lab's cages were kept. The first thing to hit me was a pungent stench of stale feces and urine. Then tiny little squeaks and scurrying sounds. Those squeaks put me on edge, and I began to tense up. Farzana pulled out 2 cages and explained that one group of animals was naive or unvaccinated, so she would be showing me how to inoculate those. The other group had been vaccinated several weeks ago, so it was time to test them to see if the vaccine had caused a significant increase in white blood cell production, a sign that the immune system is kicking in and generating virus-specific defenses. With practiced hands, Farzana held down a squirming mouse so that its belly was facing up. That's where the vaccine is injected. With one hand on the mouse, she used her free hand to uncap a test tube, load fluid into a syringe, and quickly inject the mouse. The whole thing was over in less than 30 seconds. But I was beginning to see where I might have trouble. There's an expression among people who do lab work: good hands. You either have them or you don't. I didn't. I had no trouble designing experiments and analyzing data once I got it, but often running the actual experiment took several tries due to silly things like knocking test tubes over, grabbing the wrong chemical, or forgetting to label the tubes. Now I was supposed to introduce a live, squirming animal into the mix? Right. Besides, they were actually kind of cute. I didn't say that out loud, of course. Rule number 1 amongst animal researchers: don't name your subjects. But with practice, I assured myself, I could learn to vaccinate a mouse, even ones with adorable eyes. Farzana now turned to the cage with mice that had already been vaccinated and on whom we needed to check to see if they had mounted an immune response. There are 2 ways to measure this. 1, get a blood sample and try to detect the presence of virus-specific antibodies. 2, sacrifice the mouse, slice open its belly, remove the spleen, and measure the increase in antibody-producing white blood cells that collect there. Neither one of these methods is exactly appealing. To get that blood sample, Farzana held the mouse down with one hand and used the other to apply a razor blade to the base of the tail, the part where it meets the butt. She then held a tiny plastic tube underneath the mouse, collecting drops of blood. while trying to avoid collecting other things that might emanate from that region due to the animal's stress response. People and mice are also similar in that we both piss and shit ourselves in situations like getting our asses torn apart. Farzana next demonstrated 2 options for sacrificing a mouse. A, break the neck with a quick twist, or B, slit the throat. Done correctly, either of these methods should be almost instantaneous, limiting the suffering of the animal. But both will still induce a strong stress response, with the previously alluded to shitting and pissing. Here's where I started to lose it. I didn't actually cry, but Farzana could tell I was disturbed by both of these methods. So she presented a third option: death by heat. Mice are so tiny, it doesn't take much to heat up their little bodies to fatally high temperatures. All you need to do is place them under a standard desk lamp, hold them in place. After about 30 seconds, boom, they're dead. This was somewhat more palatable. I'm not sure why. I guess because it didn't involve blood. It almost just seemed like the mice were falling asleep. Although the aforementioned stress response was still induced, and you still needed to slice open them to harvest the spleens. But for whatever reason, I decided that that would be my method moving forward. I did a few more practice runs with Farzana, each time gritting my teeth and distracting myself with conversation. I can do this, I thought. Soon enough, I'll be running my own lab, but to get there, I need to make this work. A few weeks later, it was time for me to start animal work on my own. Entering that small room alone, smelling the ambient piss, shit, and death, I knew I was in trouble. I harvested spleen on my own, twice. Both times, I was reduced to tears. After that second visit, a technician stuck her head in the door and told me that the facility director wanted to see me. What was this about? I had never actually met the director before. He just sat in his office, pushing papers, while us real scientists did the work in the trenches with the mice. I entered his office hesitantly, eager to make a good impression. Perhaps this was standard procedure? I remembered him from the humane care and treatment class, where he had struck me as a boring functionary, providing the necessary training in order to comply with funding guidelines. He simply said his staff had reported that they had observed me bent over the mice, crying, twice. He said to me, "Some people can do animal work. Other people can't. You can't." He spoke with straightforward kindness, but my heart was pounding with embarrassment and anxiety. I hadn't realized that I had been observed crying. I like to think of myself as a highly rational, as someone who makes decisions based on logic, not emotion. And this was an entirely emotional response that I couldn't control, no matter what my brain said. But I could no longer deny the visceral gut reaction that I had to the work. The facility director was right. I was not cut out for animal work. Farzana took over the animal portion of the project, bringing me freshly harvested spleens every few weeks to process in the lab. About 6 months later, we were in the midst of a challenge experiment. This means that instead of just measuring for indications of an immune response, you actually infect the vaccinated mouse with virus and see if they survive. Farzana wasn't available during the week that the observation was to happen, so I had no choice but to do it myself. Since no actual killing or blood was involved, you literally just observed the mice to see if they had taken ill. I figured I could handle it. Day 1 was fine. The mice scampered around in their cage, playing with shredded newspaper. By day 4 or 5, most were just huddled in the corner, shaking. Day 6, 3 of the 6 mice were dead, flat on their backs, legs extended in the air. Obviously, the vaccine hadn't worked. But for scientific accuracy, I had to go back and make that day 7 observation. 6 stiff little bodies, legs extended. For weeks afterwards, I woke up in the middle of the night with this grotesque image in my head. Usually, the only way to get back to sleep would be to get up and drink red wine. I sometimes envy my younger self, brimming with ambition, thinking that all I needed was a bit of intelligence and a lot of determination to succeed, thinking that I could ignore my emotions or numb them with alcohol and push through. What I failed to realize is that being a successful scientist is as much of an emotional challenge as an intellectual challenge. After the mouse fiasco, I had other struggles as a researcher. Failed experiments depressed me. I took it personally rather than letting it roll off of my shoulders, as some of my peers could do. I began to seriously question my worth as a person when I didn't make good progress in the lab. I stopped spending time on my own research and started skipping out of the lab to attend talks on origins of life research, my version of a midlife crisis, thinking I could trade in prosaic vaccine development for a sexy new topic and be happy. Ultimately, I left the lab. Even now, more than a decade later, I sometimes still feel like a failure for not making it as an academic researcher. But I have finally found success and happiness as a science communicator. Thank you.
That was Emily Burke.